Today we delve into the fascinating world of polarizability in quantum biology. So what exactly is polarizability? It quantifies how much a molecule's electron cloud can be distorted by an external electric field. In the realm of quantum biology, this concept is crucial because the interactions between molecules and external fields, like light or electromagnetic fields, determine how molecules behave in biological environments. Polarizability directly impacts optical properties, such as how a molecule scatters or absorbs light. This is super important for techniques like microscopy and biosensing. For example, in cellular imaging, polarizability measurements are used to study cell membranes or protein structures. In phase contrast microscopy, differences in polarizability between cellular components allow the visualization of live cells without staining, providing insights into cellular structure and function. Think of polarizability like the flexibility of a balloon. A molecule with high polarizability is like a soft balloon, easily deformed by pressing on it like an external field. On the other hand, a molecule with low polarizability is like a hard, inflated ball, difficult to deform. This deformation alters how the molecule interacts with light, similar to how changing the shape of a balloon changes its shadow. Next, let's explore quantum-enhanced microscopy and how it's pushing the limits of biological imaging. Classical light microscopy is fundamentally limited by shot noise, which arises because photons are detected randomly. This noise constrains the sensitivity and resolution of images, especially at low light intensities. In quantum-enhanced microscopy, squeezed light, where the uncertainty in photon detection is reduced or entangled photons are used to overcome these classical noise limits. This technique allows researchers to image biological samples with higher precision, without increasing the intensity of light, which could damage sensitive biological tissues. For instance, a quantum-enhanced coherent Raman microscope can image the vibrational modes of molecules inside living cells, allowing researchers to observe the chemical composition of cells with minimal disturbance. Quantum-enhanced microscopes have been used to study the distribution of lipids in cell membranes with a resolution that surpasses the diffraction limit. Imagine trying to see a faint object in the dark using a weak flashlight. If you increase the flashlight's brightness, you'll see the object better, but you might also damage delicate objects nearby. Quantum-enhanced microscopy is like using a high-quality, noise-reducing filter on your flashlight, allowing you to see the object clearly without increasing the brightness, thus avoiding damage. Now let's talk about polarizability as a marker of structural dynamics in biomolecules. Polarizability changes reflect structural changes in biomolecules, making it a powerful tool for studying dynamic processes like protein folding, enzyme activity, or molecular motor function. During these processes, the arrangement of atoms and electrons within the molecule changes, which alters the molecule's ability to polarize in response to an external field. By tracking these changes, scientists can non-invasively monitor molecular dynamics in real time. For example, polarizability measurements have been used to study how motor proteins like kinesin move along microtubules inside cells. As kinesin walks, its shape changes, altering its polarizability. By measuring these shifts, researchers can understand the steps of its movement and its interaction with microtubules at an atomic level. Imagine a coiled spring representing a folded protein. When you pull the spring, it stretches, unfolding. This stretching changes how the spring interacts with its environment. Similarly, as a protein folds or unfolds, its polarizability changes, providing clues about its structure and function. Finally, let's look at some quantum breakthroughs in single-molecule biosensing. Single-molecule biosensing allows researchers to study individual biomolecules, such as proteins or nucleic acids, without the need for large sample sizes. This is achieved using highly sensitive techniques that measure polarizability at the single-molecule level. Quantum-enhanced sensors, like those using quantum entanglement, improve the signal-to-noise ratio, allowing for the detection of subtle changes in a molecule's polarizability during biological processes like enzyme-substrate interactions or molecular assembly. For example, quantum-enhanced biosensors have been used to study the real-time dynamics of enzyme-substrate interactions such as DNA polymerase copying DNA. By measuring the polarizability changes as the enzyme binds to and releases the DNA, researchers can understand the kinetics and fidelity of DNA replication at the single molecule level. Imagine trying to listen to a single voice in a noisy room. Traditional methods would amplify all the voices together. Quantum-enhanced biosensing is like giving the person you're interested in a microphone while filtering out all the other voices. This allows you to hear even the faintest signals from the single molecule you're studying without interference from the noise of the environment. Probing quantum effects in biomolecular interactions. Quantum effects such as coherence, superposition, and entanglement are thought to play a role in certain biological processes. For example, quantum coherence may help proteins or enzymes transfer energy more efficiently during biochemical reactions. Measuring polarizability can provide indirect evidence of these quantum effects, as changes in a molecule's quantum state will also affect its polarizability. This could provide insights into processes like photosynthesis, where quantum coherence is believed to enhance the efficiency of energy transfer in light-harvesting complexes. In photosynthesis, the energy from absorbed photons is transferred through a network of molecules in a plant's light-harvesting complex. It is believed that quantum coherence allows this energy to travel through multiple pathways simultaneously, increasing the efficiency of the process. Polarizability measurements have been used to probe the role of quantum coherence in these energy transfers. Imagine trying to cross a river using stepping stones. Normally, you'd step from stone to stone one at a time. Classical transfer. But with quantum coherence, it's like being able to step on multiple stones at once, allowing you to cross the river faster. Measuring polarizability is like checking which stones you're on at any given time, helping scientists understand how the energy is being transferred. Dynamic polarizability in action, from theory to application. Dynamic polarizability refers to how a molecule's polarizability changes over time as its structure changes. This can be predicted using computational models based on molecular dynamics simulations, which take into account the positions and motions of atoms within the molecule. By combining these simulations with experimental data, scientists can make predictions about how molecules behave in biological systems. These models are essential for understanding complex processes like protein folding, enzyme activity, and molecular motor functions. Dynamic polarizability has been used to study motor proteins like myosin, which convert chemical energy into mechanical work. By modeling the dynamic polarizability of myosin as it moves along actin filaments, researchers can predict how changes in the protein's conformation affect its ability to generate force. Think of dynamic polarizability like watching the changing shape of a dancer's shadow. As the dancer moves, their shadow shifts, providing information about their position and movement. Similarly, as a molecule changes shape during a biological process, its polarizability changes, and scientists can track these changes to understand what the molecule is doing in real time. Quantum-enhanced Raman microscopy, revolutionizing biological sensing. Stimulated Raman scattering, or SRS microscopy, is a powerful tool that uses the Raman effect to probe the vibrational states of molecules. Quantum-enhanced SRS microscopes use quantum correlations, such as squeezed light, to improve the signal-to-noise ratio. This allows researchers to detect weaker signals with greater precision. This technique is especially useful for studying biological samples without the need for fluorescent labels, making it ideal for observing live cells in real time. Quantum-enhanced SRS microscopy has been used to study the metabolism of living cells by measuring the vibrational signatures of molecules involved in metabolic pathways. Researchers can observe how cells process nutrients or respond to drugs in real time, providing insights into cellular function and disease mechanisms. Imagine trying to listen to music through static noise. Regular Raman microscopy is like trying to hear the music with background noise interfering. Quantum-enhanced SRS is like having noise-canceling headphones that allow you to hear the music, the molecular vibrations, clearly without interference. Quantum sensors. Precision and sensitivity at the nanoscale. Quantum sensors are devices that use quantum phenomena, such as superposition and entanglement, to measure physical properties like electric fields, magnetic fields, temperature, and pressure with extreme precision. These sensors can detect changes in polarizability at the nanoscale, allowing researchers to study biological systems at unprecedented levels of detail. Quantum sensors are essential for studying phenomena like protein folding, neural activity, and molecular interactions in real time. Diamond-based quantum sensors have been used to measure the weak magnetic fields generated by individual neurons. By tracking these fields, researchers can study how neurons communicate and process information, which could lead to new insights into brain function and neurological disorders. Imagine trying to detect a whisper in a noisy stadium. A quantum sensor is like a super-sensitive microphone that amplifies the whisper while canceling out the stadium noise, allowing you to pick up even the faintest signals from the biological system you're studying. Quantum biotechnology, bridging quantum physics and biological sciences. Quantum biotechnology applies the principles of quantum mechanics to biological systems. This interdisciplinary field aims to use quantum technologies, such as quantum sensors, quantum computing, and quantum imaging, to advance biological research and biotechnology applications. Quantum effects like coherence, superposition, and entanglement enable researchers to study biological systems with greater precision, leading to breakthroughs in areas like drug design, precision medicine, and molecular diagnostics. Quantum computers have been used to simulate the interactions between complex molecules, such as drug molecules and their target proteins. By accurately modeling these interactions, researchers can design drugs that are more effective and have fewer side effects. Polarizability plays a key role in these simulations, as it influences how molecules interact with each other. Classical computers are like trying to solve a jigsaw puzzle by looking at one piece at a time. Quantum computers, on the other hand, can look at all the pieces simultaneously, making it much faster to find the right fit. Quantum biotechnology applies this quantum speedup to biological problems, enabling faster and more accurate solutions. Future challenges and opportunities in quantum biology and biotechnology. Quantum biology and biotechnology hold immense promise, but they also face significant challenges. One of the main obstacles is integrating quantum technologies with biological systems, which are inherently complex and noisy. Quantum states are fragile, and biological environments can introduce decoherence, making it difficult to maintain the quantum effects needed for advanced sensing, imaging, or computing. However, advances in quantum control, noise reduction, and quantum error correction are helping to overcome these challenges, paving the way for transformative applications in biology and medicine. One of the key challenges in using quantum-enhanced microscopes for biological research is the biological environment itself. Cells are dynamic and constantly changing, making it difficult to maintain the delicate quantum states needed for high-precision measurements. Researchers are developing new quantum control techniques to stabilize these systems, allowing for longer observation times and more accurate data. Imagine trying to use a high-powered telescope to observe stars, but the atmosphere is turbulent, causing your view to be distorted. Quantum biology faces a similar problem, where the turbulence of biological systems makes it hard to maintain quantum states. However, just as adaptive optics can compensate for atmospheric turbulence in astronomy, new quantum control methods can help stabilize quantum states in biological environments.